Welcome everybody to another video lecture. In this chapter we are looking at lipids. So, lipids are biomolecules and they contain fatty acids, steroids. Well, I guess let me be more general. Biomolecules that contain either fatty acids or steroid nucleus, which is what I'm showing you here. Lipids are soluble in organic solvents, but not in water. And they're actually named for the Greek word lipos, which means fat. These are important features in our cell membranes, in fat-soluble vitamins, in the steroid hormones, and there's quite a few classes of lipids. So we're going to describe, or the learning goal here is going to be able to describe the different classes of lipids. Now, lipids are characterized by their structures, and you can see here that there are a few different names. We have waxes, we have triacylglycerols, or triglycerides as they're also known. We have glycerophospholipids, we have sphingolipids, and we'll go over some of the variations of those. You can see here a fatty acid. This is the fatty acid structure. So a fatty acid is just a carboxylic acid with a long hydrocarbon chain, and it's the long hydrocarbon chain that makes this not soluble in water, that gives it the fat properties. So triacylglycerols or triglycerides and glycerophospholipids, so two different types, these both contain the alcohol glycerol. So that's one of the distinguishing features of this category or class. So just as a quick reminder, fatty acids are going to be esters. The triacylglycerols or the glycerophospholipids or sphingosine, even if we look at that one, they're esters that when hydrolyzed yield fatty acids and alcohols. There's going to be a few types of fatty acids. We'll look at those. There's going to be a few types of alcohols, whether or not they are glycerol or amino alcohols, and we'll go over those differences. Or even sphingosine. Sphingosine is going to have an amine functional group on it. We haven't looked at amides yet, but that's this bond right here. It's the ester. The fatty acid itself, we saw here, has that carboxylic acid functionality. And glycerol, which we haven't seen yet, but I can show you with just a real general. If this is glycerol, it's actually a triol. Glycerol has this structure. So this guy. And so what we see here is the fatty acid, the fatty acid part and the alcohol part react together to give that bond. And we'll go over this a little more down the road. So these are the glycerol-containing lipids, the triacylglycerols and the glycerophospholipids. We also have sphingosine-containing lipids, and we call those sphingolipids. And you can see that what differentiates these. Now these of course aren't structures yet. We're just using boxes with names in them, right? We'll look at structures here in a second. But what this is supposed to represent is that it's kind of like glycerol with a fatty acid already built onto it. This is also an alcohol, this sphingosine. So these would be sphingolipids. And then we have steroids, and of course there's waxes as well, which I don't think I've shown on here. I'll show you a structure of a wax here in a second. Steroids, which are completely different in structure from the other types of lipids, they don't contain fatty acids and they can't be hydrolyzed. So no fatty acids, no hydrolysis. They're characterized by this steroid nucleus. It's 4 fused carbon rings. Pay attention to how A ring and B ring connect together and how C ring connects to B ring. It isn't a straight line. And then the D ring here has 5 members, where all of these have 6. So in general here, you've got the organization here. Naturally occurring compounds, again, in our cells and in our tissue, soluble in organic solvents, but not soluble in water. Waxes are on this table, so long chain alcohol and a fatty acid. Don't know if I have any pictures of waxes though. Let me draw you a wax. So a wax would look something like that. So difference is the long chain alcohol is also very nonpolar, where glycerol is a real short chain alcohol with our fatty acid. So waxes have that ester group in them and 2 long hydrocarbon chains that make them very insoluble in water. They offer protection to fruits and stuff like that. I don't know if we talk about it too much. There are more important biologically relevant lipids that we go over. All right, quick learning check. Go ahead and pause the video, give it a shot. Carboxyl groups and long carbon chains are on fatty acids, fused 4-member ring, steroid. All right, another one. Which one of these is not characteristic of lipid? Should be easy. They are not soluble in water. We might think of saturated fatty acids as chips with regular shapes, so like Pringles. They stack closely together. Similarly, we may think of irregularly shaped chips, which these are still the same shaped chips, but let's pretend that they're irregularly shaped so they can't stack together. These would be like unsaturated fatty acids, where these would be the saturated. So our learning goal here is going to be draw condensed structural and line angle formulas for fatty acids and to be able to identify if it is saturated or unsaturated. So fatty acids, simplest types of lipids, long unbranched hydrocarbons. They do have the carboxylic acid group at the end. Between 12 and 18 carbons long, typically. Insoluble in water because of that many carbons. Saturated means they do not contain a carbon-carbon double bond. Unsaturated means that they do contain carbon-carbon double bonds. So let's take a look at some examples of how we represent these. So in the line angle structural formula of the fatty acid, the ends and the bends of the lines are all carbon atoms. So line angle, much easier to draw. Remember, you just count the corners. Condensed structural formula, you'd show everything. All the internals are CH2s. And then your carboxylic functional group. You can condense that and just take all your CH2s. So here we would have 10 of them. And you could just put parentheses around it and say 10. And you can even condense the carboxylic acid functional group to be COOH. So shortest way to write a fatty acid would be like this. We tend to show them like this. And of course the ball and stick model. So you can see everything. This is lauric acid. Now, how do I know it's lauric acid? Because that's what it's named. When you have 12 carbons, it's lauric acid. Notice the notation here, 12, 0, 14, 0, 16, 0, 18, 0. The 0 here represents the number of double bonds. So since these have no double bonds, they are saturated. They have the maximum amount of hydrogens that they can have. Notice that they're also evenly numbered. This has to do with how they're formed. It's very rare to find an odd numbered fatty acid. These are present in different substances. Most of these look like they come from plant substances, and then we've got stearic acid here, which comes from animal fat. I forget what stearic means. I feel like it means fat, or it means meat, or something like that. Never mind. Um, okay, so these are. I'm almost remembering that this used to mean something. I forget how this was named. Anyways, of course, it's not important. So melting points, that's what I'm pointing out here. So what you can see here is with the melting points, as the carbon chain gets longer, the melting point increases. This is because the longer the carbon chain, there's more interactions between neighboring fatty acids. All these little spots where they can attract each other create a situation where you need a higher temperature to get them to split apart. If we have short chains stacking together or being influencing each other, that takes less energy. Let's look at some monounsaturated fatty acids. So these can be double bond containing. So 16,1, 18,1. Just to go back, 16 was palmitic acid. When we add a double bond to it, palmitic becomes palmitoleic. And then stearic acid becomes oleic acid when we add a double bond to it. So you don't need to know these names. Just be aware that different numbered carbons and different numbers of double bonds usually get their own name. Palmitoleic can be found in butter, oleic acid in olives, pecans, and grape seeds. The double bond is often shown in its cis format. Trans double bonds are something different we'll talk about. In the cis format, this creates a kink in the hydrocarbon chain, makes it so that it's an irregularly shaped chip that doesn't stack together well anymore. This lowers the melting point. So let's jump back real quick. So you can see here, if we take a look at palmitic and stearic, we have 63 degrees and 69 degrees respectively for their melting points. So you gotta heat these up to get them to melt. Palmitoleic is 0 degrees. So it's liquid at the same temperature water is liquid. Oleic acid, 14 degrees Celsius. These require less energy to melt because they don't pack together as nicely, and so they don't keep each other in liquid state as easily. We can have polyunsaturated fatty acids. They contain at least 2 double bonds. So linoleic here would be what oleic acid becomes. So again, stearic acid, then oleic, and now linoleic, as it has 2 double bonds. I think it went from 14 degrees now to -5. Linolenic, -11. Arachidonic. We'll talk about this when it comes back. Maybe it isn't in this class. I forget which class I teach that we talk about arachidonic acid, but it is a biologically relevant molecule. I think we do talk about it here. Or maybe it's when we get to hormones. I think that's here. Anyways, this one has 4 double bonds. We can even start numbering where the double bonds are from the omega side of the carbon chain. So whenever it is on the number 3, we call these omega 3. When it's on the 1, 2, 3, 4, 5, 6, that'd be an omega 6. 1, 2, 3, 4, 5, 6. That's also an omega 6. So omega 3, omega 6, omega 9 fatty acids refer to the position of those double bonds when counting from the end of the chain. Alright, so here's a little graphic to show you how that cis kink actually leads to a pretty dramatic shape change in our molecule. Of course, the trans isomer does still maintain a normal straight chain without a kink. Almost all naturally occurring unsaturated fatty acids have one or more cis double bonds. All right. Now, just to kind of go over it one more time, when they contain only single bonds, they pack together. That means they have strong dispersion forces, and it takes higher temperature to get them to melt. Comparison here now showing you the graphic with the chips packed together. So these are all single bonds, 69-degree melting point. These guys are all cis bonds, specifically also kind of jumbled around, because you could be like, well, why aren't they all just like packed like this? They could be, I suppose. But here, they have a much lower melting temp because of the double bonds. All right, quick learning check. Consider this line angle formula for Vazquez acid and give some answers to the following questions. So, why is it an acid? How many carbons does it have? Saturated, mono, poly? Is it liquid or solid at room temp? What would you think? And do you think it'd be soluble in water? So pause it, give it a shot. So it's got the functional group for carboxylic acid. It's got 18 carbons. It's a monounsaturated omega 9. 1, 2, 3, 4, 5, 6, 7. Omega 7. Liquid at room temperature, because I guess room temperature is around like what, 25? So this would, if we go back and look, one double bond usually lowers it to right around 0. Would it be soluble in water? No. Too many carbons. All right. Humans are capable of synthesizing some fatty acids from carbohydrates. There are others that we cannot synthesize. So we have to get them from our diets. These are the ones that are known as the essential fatty acids, the ones that we have to get from our diets. Those include linoleic, linolenic, and arachidonic. Prostaglandins. These are the hormone-like substances. They have to do with, I think there's some pain responses in here maybe. Let's see. They're also known as eicosanoids. They're formed from arachidonic acid, that 20-carbon polyunsaturated fatty acid. We're going to see that they have some differences. We're going to look at a few different prostaglandins and just to see how they have the same sort of parent chain and then can have different effects based on their different substituents. Lots of different functions, lowering or raising blood pressure, stimulating contractions and relaxation of smooth muscles. So different substances, different effects probably on depending on where in the body they are, and again what variation we have. So here you can see prostaglandin E and prostaglandin F. Prostaglandin E has a ketone group on carbon number 9 on this 5-member ring, where prostaglandin F has a hydroxyl. And the number of double bonds, I think, is also different, the same? I guess here it's the same. What they're showing here in this number is the number of double bonds. That's what this little 1 is representing. Here's a few more. Or actually, what is this? This is, this is kind of showing the original molecule, arachidonic acid. When tissues get injured, arachidonic acid that you have in your body gets converted into prostaglandins that produce the inflammation and pain response in that area. So depending on what area of the body or what prostaglandin it's going to be converted into, it'll get different modifications, whether it's that ketone group or I guess in this case, well, I guess we don't really know what all of these do. There's a double bond here. That's a variation from this one. So yeah, from arachidonic acid, we can generate the other prostaglandins. Now, prostaglandins can be blocked from forming using non-steroidal anti-inflammatory drugs or NSAIDs. These block the production of prostaglandins, and by stopping the conversion of arachidonic acid into these compounds, they effectively decrease pain and inflammation. Some of the NSAIDs include naproxen, like Aleve and naproxen, or ketoprofen, which is Actron, and nabemutone, relevant. All right. Let's see. Unsaturated fats, such as those in vegetable oils and fish, they're actually recognized to be more beneficial to health. I was alluding to the naming of these earlier. Vegetables that contain omega-6 fatty acids, in particular, um, seem to be really beneficial. And again, we name these from the methyl end of the chain. And so, linoleic is an omega-6 fatty acid. Arachidonic acid is an omega-6 fatty acid. Fish have really high levels of omega-3 acids. So that just means that the first double bond appears at the carbon number 3 from the methyl end. And omega-3 acids include linoleic, eicosapentaenoic, docosahexaenoic acids. I mean, there's a ton. You don't need to know all the names of those. But yeah, cold water fish are a source of the omega-3 fatty acids. Um, Lower the tendency of blood platelets to stick together and reduce the possibility of clots are just one of the things that we see as a benefit for the omega-3s. This is particularly important if you suffer from atherosclerosis or heart disease, which can make it difficult for your blood to circulate. All right. Learn and check. Label each of the following acids as either saturated, monounsaturated, or polyunsaturated. Pause it, give it a shot. All right, so you would obviously have to look these up. I don't expect you guys to know them by name, like whether or not they're unsaturated or mono or poly, um, or saturated, but you should be able to look up the structure and be able to give me one of these names, okay, and know what it means and that sort of thing, or to give me the notation, of how many carbons it has and how many bonds, to know what that means. If I were to give you this notation, maybe you could pull up this name, but I don't tend to ask those kind of questions. All right, I'm gonna have to continue this in part 2 because I have a screaming child.